My name is Helena Moreira. I'm a researcher in Universidade Católica Portuguesa in Portugal, and I work with bioinocula in phytoremediation or in sustainable agriculture, in phytomanagement as well. So today I'm presenting two studies of phytomanagement of metalloids contaminated soils. I'm going to give you an overview of what is phytotechnologies, what is phytomanagement, what is metalloids, the contaminated soils, and what are the problems that we have in producing bioinocula in the lab, and the problems that we have when we are going to a contaminated site and we have to see the pollution, to see what kind of plants are going to produce, what kind of bionocula is the most suitable. So I'm going to talk to you a little bit about these things. You are welcome to make questions whenever you want. I can stop the presentation and answer. Of course, you can. You are most welcome to do that. I'm sorry. So soil pollution is a problematic issue worldwide because soil is a non-renewable resource, and its pollution impacts not only the ecosystems but also the human health. And the pollution of soil is impacted by agriculture's practices due to the overuse of fertilizers and pesticides, waste disposal, commercial activities, transport spills, and of course the industrial and mining activities. I'm sorry. I'm sorry, it's my mobile phone. And the soils are polluted either by organic and inorganic substances. I work mainly with trace elements, metals and metalloids, but we can find in soils a mixture of organic and inorganic pollutants. Not only we find the soils with metalloids and non-metal substances, but also with PCBs, with benzene, with dioxins, a lot of substance we can find in the soil. And why should we think in remediating soil? Why should we think in recovering soil? Because soil has several functions. Soils deliver ecosystem services that enable life on Earth. For instance, carbon sequestration, provision of food, fiber and fuel, climate regulation, nutrient cycling. So it's very important, the functions that soil performs to humankind. So this is better if we use the sustainable technologies to recover soil pollution. And this has been a research, an issue in research all over the world. And it has been also an interest by policymakers to make legislation to face this problematic issue. So why we should use phytotechnologies, and what are phytotechnologies? Phytotechnologies are the use of plants and associated microorganisms to extract, degrade, contain, or immobilize pollutants, and can be performed in soil, not only in soil, but also in groundwater, sediments, and other contaminated media. And how we can do that? We can use phytoextraction and phytostabilization. I'm going to talk a little bit about this in a while. And why it's better than the civil engineer techniques? Because it's less invasive, it's cost effective, improves soil health and functions, and prevents erosion and pollutants leaching. And the technologies that I was talking about that are mainly suitable to trace elements are phytostabilization, where trace elements are stabilized in the roots of the plants or in the soil through precipitation, or onto absorption in the roots or inside the roots, or by phytoextraction, where the pollutants are taken up by the plants and the plants take up to the harvestable parts. So they translocate the metals from the roots to the shoots. and the amount of trace elements is decreasing with the time in the soil. In the last few years, a new concept arose, which is phytomanagement. And phytomanagement allies the environment and the economy. And the stakeholders, the site owners, can benefit from environmental revenues, which are the increase of biodiversity, the protection of the soil, the carbon sequestration, and other environmental benefits that I was talking about, but also from economic revenues, because they can use those soils to produce biomass. And that biomass can be used for the production of timber, to be used as feedstock for pulp and paper, adhesives, etc., can be used to grow some bioenergy crops, to produce biodiesel, bioethanol. So they can be used for several things, and stakeholders can get some money from those contaminated sites. Those sites can also be used for phytomining, which is the extraction of valuable metals such as nickel from the plants, and can be used also to produce some fortified crops, which is enriched with trace elements such as selenium or zinc. But what we have to consider in phytomanagement, we have to consider a lot of stuff. The plants are not only the players. We have, besides the plant, the cropping system, which is the organization of the plants, the cropping patterns. Which amendments should we add to the plant's drive? And what bioinocula could we use to promote plant growth and resistance and resilience to the trace elements present in the soil? So what criteria should plants fit? So they should be easy to grow and harvest. They should yield high biomass. They should be, of course, pollutant tolerant, and they should be fast growth rate. And we have several plants within these concepts, such hemp, such maize, such sunflower, tobacco, miscanthus, and several trees like poplar, willow. So there are several types of plants that we can use. Cropping systems, what are the cropping systems? So we have to consider the climate conditions of the site. So the crops have to be tolerant to that climate conditions. We have to think if we want annual crops, perennial crops, if we want to put some cover crops to stabilize the soil, to decrease the soil erosion and decrease the trace element leaching. We should design and monitor all the things that we do in the field. The sequence of the crops have to be thought. So the cropping patterns, all the management stuff. So we have to think in a lot of things when we are thinking in the phytomanagement of a trace element contaminated soil. Sometimes we have to use amendments to recover the soil because the plants cannot grow without these amendments. Sometimes it's possible, sometimes we cannot do it without these amendments. And there are several types of amendments. We have the manures, the biochar, compost, green residues, biosolids. It always depends on what we want, what we have available. So it depends on many factors, but there are a lot of choices that we can make. And then we have the bioaugmentation, which is the use of bioinocula, the use of bacteria, the use of fungi that can help plants thrive in those harsh areas and can help also the mobilization or immobilization of the trace elements in the soil. And in bacteria, we can inoculate one strain, we can inoculate several strains, the same with the fungi, single strain, a consortium of fungi, or a composite bioinocula with bacteria and the fungi. And what kind of this microorganisms service provide? So the bacteria, we usually use the plant growth promoting bacteria. There are several types of plant growth promoting bacteria, and they can help plants with growth promotion. They produce growth hormones like auxins, gibberellins, cytokinins. They release stress reducing enzymes and they induce the systemic resistance of the plant. And they also help the plants to acquire some nutrients like phosphorus and potassium They help plants to fixate nitrogen, and they also release siderophores that are molecules that chelate iron and then they give to the plant. And they also have the biocide production, which prevents the spread of diseases to the plants. And we can have also fungi, and within the fungi, we have the mycorrhizal fungi that associate intimately with the roots of the plants. And it reduces also the plant stress, improves the water and nutrient acquisition by plants, and promotes the plant growth. And besides that, in the soil, it decreases soil organic matter, it reduces erosion, and reduces trace element lixiviation. So together, they will improve the phytomanagement in the efficiency. That's what we want. But for this inocular selection and production, we have a stepwise process that begins in the isolation and characterization of bacteria in this case, but we have also the isolation and characterization of the fungi as well. They have to be, of course, non-pathogenic, both for plants and for humans. They have to be easy to produce, adapt to the several types of environment conditions, because when you're going to inoculate it in the field, they are going to face sometimes very low pH, low nutrient content, so they have to face very harsh conditions. And we have then to describe their mechanisms of actions, their traits, why they are important, if they produce high levels of growth hormones, if they produce ACC-deaminase, that reduces the ethylene levels, which is a stress hormone. So we have to describe very well this bacteria. And then we go to the greenhouse and make some combinations between the bacteria or between the bacteria and the fungi. And we test in different soils because they can act and be effective in a specific soil. When we go to the other soil, they don't work at all. We have to test it with different hybrids and cultivars of the plants. And we have also to think in the number of cells that we are going to inoculate. But a little bit amount of cells may not have the efficiency that we want. So we have to see which inocular size is the best for that kind of soil and that plant. And then we go to the field trials where the plants and the bacteria or the fungi, our inocula, have to face several types of edapoclimatic conditions, trace elements, not only one, but sometimes a multi-contaminated area is available and the bacteria have to thrive there. And sometimes they have to face drought and salt stress, several abiotic and biotic stress they have to face. So we start with the greenhouse studies, as I told you, and most of the times the inocular experiments to see whether inocula is effective or not, we test it in the sterile soil. Because in a sterile soil, we don't have the native microbial populations. And without the native microbial populations, we only see the efficiency of bacteria. But the things can be very different when we have the native microbial populations. And that's what we wanted to know, to see in this lab study. So we picked up sterile soil and we compared with non-sterile soil. So inoculated bacteria in sterile soil and in non-sterile soil, and we compared between both. Also, a thing that is very common is to spike the soil with one type of trace element. And this is also very different from what we face in the field, because the field are naturally contaminated. The absorption to the soil particles is different. The maturation is different of the trace elements in the soil. Normally in the soil, we don't find only one, but several types of trace elements. So the conditions are very different. So we wanted to test if our inocula were really effective under sterile conditions, but were also effective under non-sterile conditions. And for that, we spiked sterile soil, and we picked up field contaminated soil, and we make it the sterilization as well, to see if the performance was the same, if there was some consistency between the results, or we have some overestimation of our bacteria in sterile conditions. So we want to test the efficiency of the two plant growth promoting rhizobacteria to promote the growth, in this case, zinc accumulation in maize plants, in spiked and sterilized conditions, and we wanted to compare with field contaminated under sterilized and non-sterilized conditions. We wanted to evaluate the transferability of the results and determine whether the soil conditions affect the performance of bioinocula. So we picked up two PGPR, Cuprea avidus necator and Acrisium bacterium umi. We made the traits available. So we searched for EIA production, HCN, ACC deaminase activity, ammonia and siderophores. We have already tested these bacteria in other conditions, and we saw that they were able to increase the growth and nutrient uptake in maize grown in non-contaminated soil for agriculture's purposes, and in soil contaminated with cadmium. These two PGPR, I'm sorry again, were retrieved from Estareja Creek, which is a chemical complex here in Portugal, and was retrieved from the sediments with trace elements in the lab. So we made two experiments. The experiment one, we picked up some agricultural soil, was sterilized, and we spiked with chloride zinc to achieve the final concentrations of 100, 500, and 1000 ppm of zinc. Then we inoculated both bacteria in maize, and we determined biomass, the zinc content, and zinc bioavailability. In experiment two, we retrieved the soil from the industrial chemical complex of Estareja. We sterilized the soil, and we put some non-sterilized soil as well to perform the same experiment. And the soil has more or less 600 ppm of zinc. And here we inoculated B1, B2, and a mixture of both bacteria. And we determined the same parameters, which were biomass, zinc content, and zinc bioavailability. Regarding the experiment one, what we saw is what we expected. There was a decrease of root biomass with increasing zinc concentration, but PGPR were able to increase root biomass. And Acrisium bacterium umi was a little bit better of comparing with the Cuprea avidus necator, which was the B1. Regarding shoots, there was a slight decrease of shoot biomass with increasing zinc concentration, and the PGPR were able to generally increase the shoot biomass. But the performance was similar between Acrisium bacterium umi and Cuprea avidus necator. Regarding zinc accumulation, there was an increase of root and shoot accumulation with increasing zinc concentration. This was expected as well. PGPR was able to increase zinc accumulation in roots at 100 ppm. And generally, PGPR increased zinc shoot accumulation, especially the bacteria B2. Regarding some criteria, some indicators that we have to see if the phytoremediation is being well done or not, we have the bioconcentration factor, which is the ability for plants for elemental accumulation from the substrate. And we can have the elements concentrating more in root or in shoot. In this case, we have the bioconcentration factor above 1 in root and below 1 in shoot. And we saw that bacterial inoculation could even increase the BCF values in roots. And Acrisium bacterium umi increased the bioconcentration factor in shoots in all zinc spiked soils. Regarding the translocation factor is another indicator and tell us the plant's potential for elemental translocation from roots to shoots. And this was below 1, which means that the metals the metal were concentrating more in roots than in shoots, and there was no effect of bacteria inoculation. What we saw also regarding the zinc bioavailability in soil is that the zinc bioavailability increased with increased zinc concentration in soil, and bacteria had no effect. Regarding the experiment 2, that we retrieved the natural contaminated soil, we saw that there was lower shoot biomass in non-sterile conditions, and the inoculation with the bacteria increased biomass, especially under sterile conditions. Regarding the zinc accumulation, there was a higher zinc accumulation in non-sterile soils, which were related with the lower biomass, and there was an increased root zinc accumulation in non-sterile soils for plants inoculated with the plant growth promoting rhizobacteria. And there was no observable effect or a negative effect of PGPR inoculation in shoot tissues in both soils elements. Regarding the bioconcentration factors, in sterile conditions were below 1, either both in shoots and roots, but the metals were again more concentrates in roots. And bionocula did not significantly affect BCF in roots in this case, and generally decreased the BCF in shoots. In non-sterile conditions, there was a bioconcentration factor above 1 in roots and below 1 in shoots. And bioinocula were able to increase BCF values in roots and decrease in shoots. Regarding translocation factor in sterile and non-sterile soils were below 1. There was no effect of bacterial inoculation. Regarding zinc bioavailability, the extractable zinc was lower in sterile soils and higher in non-steriles. And bacterial inoculation decreased extractable zinc in sterile conditions and increased in non-sterile conditions. So what we see is the PGPR inoculation improved maize biomass in zinc spiked soils as well in field contaminated soil, but sterilization strengthens this effect. So we have higher biomass in spiked soils, and for field contaminated, the sterilization still have an outcome of higher biomass than in non-sterile conditions. But bioinocula induced different zinc accumulation patterns in spiked and field contaminated soils. And what we saw also was that sterilization reduced zinc bioavailability and affect the plant accumulation patterns. Maybe it was because of the extraction of soil aggregates that increased the clay's fraction, and the increasing soil clay's fraction, there was new reaction sites to increase metal absorption, but it is supposing, we don't know for sure, we had to test this hypothesis. So what conclusion do we get? The bioinoculant maintained the positive effects, but the sterilization affect bioinocular performance and metal accumulation due to the removal of microorganisms native to soil. And sterilization affect bioinocular performance and metal accumulation due to changes in metal availability. So these screening experiments lead to different performance, lead to overestimation, and are inconsistent with the results that we can have in the field conditions. So we should do these tests in greenhouse tests with contaminated soil and without sterilization, so without spiking the soil as well. Also, what we saw that maize is a good candidate for phytotechnology solutions, especially for phytostabilization, because there was a higher accumulation in roots than in shoots. And this PGPR was beneficial plant growth promoters, so we can go through to fill the contaminated sites and test this maize, this variant of maize, and this bacteria. And now I'm going to present a field study that we performed under a project that was a PhytoSudWare project, which involved 3 countries. It was Portugal, Spain, and France. And this project was to demonstrate the feasibility of phytotechnology across several types of edapoclimatic conditions. And we worked specifically in a mine, Borralha mine, here in the northern Portugal. And the Borralha mine was a mine of tungsten for tungsten production. It still has the tailings and ponds exposed to climatic conditions. So there is wind and aeolic and water erosion that affects all the surrounding soils. And the soil has higher concentration of copper, zinc, cadmium, arsenic, and lead. And here is our plots established in the area within the mine boundaries. And the people here in the vicinity plant the maize, the lettuce to eat. So they eat vegetables grown in soils contaminated with this high concentration of trace elements. So we wanted to test if you can do phytomanagement in this soil. And we planted sunflower. It chose sunflower and poplar because these plants were suitable for those climatic conditions. And we tested under 700 square meters. And poplar and sunflower cropping patterns. Poplar was intercropped with a leguminous, which is alfalfa, and they were inoculated with arbuscular mycorrhizal fungi and plant growth promoting rhizobacteria. Sunflower was also under a cropping pattern, which was winter cropping. So during the winter, we planted clover and alfalfa to enrich the soil with nutrients, and during the summer, we plant sunflower. And we also inoculated the sunflower, part of the sunflower, the other part was the control, with arbuscular mycorrhizal fungi and plant growth promoting rhizobacteria. And this trial was in the field during 2 years. And we still are analyzing the results, the several parameters, because there were several types of measurements that we had to make. The plant's establishment, the biomass that they achieved, the trace element concentration in the tissues, the trace element availability in the soils, the nutrient content of the plant, the physiological status, the soil enzymes, if there was an improval of soil enzymes, the soil microbial communities, if there was a change in the microbial communities in these soils under phytomanagement, or there was no change at all. So these things all have to be evaluated to see if the phytomanagement is feasible in a certain area. So here's the sunflower, some pictures from the beginning of the experiment, here at the end of the experiment. So it was able to establish and grow. So, but what we saw, it's the non-inoculated sunflower had higher biomass than the inoculated ones. And we found that the inoculated plants had higher concentration of trace elements. So they were able to phytoextract elements, and that's why they have lower biomass. But even though they were able to grow, to have a good biomass, and to establish in those higher contaminated sites. We see it through these indicators, the bioconcentration factor and translocation factor, that the inoculated plants had higher bioconcentration factor, either in both in roots and shoots. The trace element bioavailability was also higher in inoculated plots. So that's why the plants were able to take up more trace elements for their tissues. We also saw that they have a higher content of nitrogen and phosphorus, so the bioinoculation were able to increase the nutrient content of these plants. But other physiological indicator told us that the plants were under stress, the plants inoculated. That's because they had higher concentration of metals. We still are analyzing the bacterial and fungal diversity. We see that we have some differences in bacteria and fungi, but we still didn't gather all the results and make the correct analysis to see what are driving these differences. We also have the bacterial and fungal composition. We see that there are some differences between unplanted, inoculated, and non-inoculated plots, but we have to figure it out. Why are these differences, and are these differences significant or not? And we have a lot of data regarding the prokaryotic and fungi diversity. And we have to establish some correlations between also the trace element concentration, the bioavailability at the end of the experiment. So we are still working in this data to see if really the phytomanagement works under this precise trace element contaminated soil. The poplar is still under analysis because we still have the poplar on those sites growing. And now in October, we are collecting the final samples to analyze and then to collect all the data and analyze in an integrative manner. So thank you very much for listening to me. I'm able to answer to all your questions.
Thank you too, very interesting and useful presentation. Thank you, dear Helena.
Thank you.
Thank you, Helena. It's a very good idea for our local research.
Oh, yeah. Thank you. It would be great if we could work with each other.
Thank you, Helena. I consider today's webinar is timely, actually, and informative webinar. Thank you.
Thank you. Thank you.